This video is going to continue our conversation on eukaryotic cells, and we are going to focus on three types of organelles specifically: the lysosome, peroxisome, and vacuoles. So lysosomes are key for digesting stuff. That stuff may be pathogens. It could be organelles that don't work anymore, okay? So whenever there's something in the cell that needs to be broken down and eliminated, the lysosome is called to play. So we just say kind of vaguely that its function is digesting. And how it actually digests things is through the use of hydrolytic enzymes. So remember, enzymes are specialized proteins. They have a specific job and they do it well. These particular enzymes break things down. So you'll notice that the lysosome has a variety of hydrolytic enzymes, really so that it can break down just about anything that it needs to. But these hydrolytic enzymes only work in an acidic environment. Therefore, the environment of the lysosome itself must be acidic because it needs these hydrolytic enzymes to work. So if you look at this top picture over here, you are going to notice that in this case, in this case, the cell has taken in food from outside of the cell. It's holding on to this food in a little vacuole. And the lysosome is coming in here. It's going to fuse with this vacuole and expose those little blue hydrolytic enzymes to the food, because it's going to break down that food into little pieces and extrapolate nutrients from it. So as you can see, once those hydrolytic enzymes gain access to the food, it breaks it down, and then the cell can use it as it needs. So one thing I want you to think about is the importance of this acidic pH. Not only is the acidic pH required so the hydrolytic enzymes can work, but this also serves as like a safety feature for the cell, because overall, the cell is much larger than an individual lysosome. And the overall pH of the cell is neutral. And this is important because what happens if we have a leaky lysosome, right? If we had a leaky lysosome, then those hydrolytic enzymes would escape from the confinements of the lysosome and get out to the cell. That could be problematic, right? We don't want hydrolytic enzymes breaking down functional organelles. So because hydrolytic enzymes require an acidic pH, should there be a leaky lysosome, the rest of the cell is protected because those hydrolytic enzymes wouldn't be able to work in the neutral pH environment of the cell. Okay, so it's important that only the lysosomes have that acidic pH. That way, the hydrolytic enzymes only work when they're in that environment. All right, the peroxisome is responsible for detox on a much larger scale than the smoothie R that I had mentioned before. So the peroxisome is going to degrade poisonous and metabolic waste. And so one way it does that is actually by creating harmful intermediates. So when we think of oxygen, we think of O2, right? What we breathe in, what we need for survival. But there are actually more dangerous forms of oxygen. And some bacteria are anaerobic, so they can't handle any form of oxygen. We're going to talk a lot more about that in Unit 2. But sometimes during metabolism, some of these dangerous forms of oxygen are produced. And the cell has to be able to handle that so it can continue on, doing metabolism and surviving. And so the peroxisome has certain enzymes that help handle these toxic forms of oxygen. One is oxidase and one is catalase. And we're going to talk about these in lab. So I'm just going to touch on them real quick. So catalase, let's just kind of focus our attention on that one for right now. Catalase is able to break down hydrogen peroxide, which is H2O2, into water and gaseous oxygen. So if hydrogen peroxide is produced as an intermediate, then catalase will quickly convert it to water and gaseous oxygen, which that cell would be able to handle. Okay, so sometimes though, a hazardous molecule will be encountered, and in order to handle that toxic molecule, it will actually be converted to peroxide. But then we just talked about how peroxide can then be converted into safer molecules for the cell. So the peroxisome is able to handle all these toxic molecules because it has its own staff, its own little workers like catalase and oxidase that are going to take those harmful things and turn them into safe molecules. And then lastly, for this video, we're going to talk about vacuoles. And vacuoles are really like storage bins. They temporarily hold on to things that the cell needs. We saw a vacuole earlier in this video in that top picture where the vacuole is just holding on to food. Plants actually have what's called the central vacuole, which you see on that bottom picture. And the central vacuole holds on to a lot of water, right? That way it can store water in case the plant cell finds itself in needing some water. And so just think of vacuoles as little storage facilities. It holds on to whatever the cell may need. All right, y'all, that is it for this video. Let me know if you have any questions.